Okay, so if you've ever, looked in the mirror and thought maybe there's a bit less hair than there used to be, you've probably come across treatments like platelet-rich plasma, PRP. It gets talked about quite a lot, right? As a lot promising thing in regenerative medicine, a potential answer for hair loss. But what happens when someone takes a really close look at the actual data behind one of these studies, like really digs in? And what if that closer look finds some, well, pretty surprising numbers that make you question the whole basis of the study? That's exactly what we're getting into today. We're unpacking a specific letter to the editor, and the title alone tells you something's up. It's called The Emperor Has No Platelets. I mean, the Emperor has no platelets. You gotta love that.
Yeah, it's pretty direct.
It immediately signals this isn't just some dry academic back and forth. It hints right away that maybe the key ingredient in this treatment, or maybe it wasn't what everyone thought. So by the end of our deep dive today, you'll get a much clearer picture of why it's so critical to look past the headlines, past the reported results in scientific research. And you'll see how digging into the data, like really digging in, can tell a completely different story. Okay, let's do this.
Right. So the story really starts with the original study that prompted this critique. It was by Dr. Gordon Sasaki, and he was investigating PRP for treating alopecia, that's, hair loss. Specifically, he compared two different supposed platelet concentrations of PRP against a placebo.
Okay, so comparing different strengths of PRP to see what worked best.
Kind of. His study did find some association between improvement and what he called the higher PRP concentration. But, and this is important, it didn't reach statistical significance.
Right, meaning the results weren't strong enough to rule out chance.
Exactly. So you couldn't definitively say the PRP caused the improvement based on his published results.
Got it. Now, you mentioned PRP concentrations. For listeners who might know the term, but maybe not the specifics, what actually defines platelet-rich plasma? Is there a threshold?
Yeah, there is. Classically, PRP is meant to have a minimum concentration of 1 million platelets per microliter. That's the benchmark.
1 million per microliter.
Okay. And on the flip side, you've got platelet-poor plasma, or PPP. That's when the concentration is below normal whole blood levels. Typically, that's around maybe 200,000 platelets per microliter, sometimes less.
And then there's this other term, PIF, platelet increase factor. How does that fit in?
PIF is crucial here. It's simply the ratio. It compares the platelet concentration in the final PRP product to the patient's baseline whole blood platelet concentration.
So it tells you how much the platelets were actually concentrated.
Precisely. And Dr. Sasaki's study reported a PIF of 4.5x. Now here's a key detail. That 4.5x number, it came from the manufacturer's datasheet. The manufacturer was Eclipse, and they also happened to sponsor the study.
Okay. Manufacturer data, and they funded it.
Right. And this 4.5x PIF was widely seen, and frankly still is by many, as sort of the optimal level for getting good results with these kinds of treatments.
So that's the backdrop. Manufacturer claims 4.5x, study uses it, results are kind of meh, statistically speaking. But then along comes Dr. Patrick Yam.
Yes, a clinical instructor. And he decided to look much deeper into Dr. Sasaki's study, specifically at that platelet concentration claim.
And what did he do? Did he just question the theory, or?
No, he went back to the data Sasaki himself had generated. See, Dr. Sasaki had actually sent 1 samples from each batch of PRP he prepared to a hospital lab. They used what's called a culture counter analysis, or CCA.
Okay, what's a culture counter?
It's basically a standard lab machine, very accurate for counting cells like platelets in a fluid sample, so it gives you a precise number.
So Sasaki got these CCA results back to verify the concentration.
Yes, and in his paper, Dr. Sasaki stated that the CCA results confirmed the 4.5-fold increase. He said they confirmed it.
Okay, sense a but coming.
A big but. Dr. Yam took that same CCA data reported in the study, and he just did the math. And what he found was, shocking.
Don't leave us hanging. What did the math actually show?
Okay, so let's take the male subjects, using what Sasaki labeled Batch A PRP. The culture counter data, Sasaki's own data, showed a mean total platelet count of about 137 million platelets in the 5 ml sample volume used.
Right.
Now, when Dr. Yam converted that total count into a concentration platelets per microliter, it came out to just 27,398 platelets per microliter.
Wait, 27,000? You said the minimum for PRP was 1 million.
Exactly. And remember, the average baseline platelet count in whole blood for these subjects was around 276,750 per microliter. So Dr. Yam calculated the actual platelet increase factor, the PIF, based on Sasaki's own lab results. It wasn't 4.5x, it was 0.1x.
zero point 0.1. That's not an increase at all. That's a massive decrease from baseline blood.
Precisely. It's a tenth of the baseline concentration.
So the concentration used, that 27,000-ish number, how far off the supposed optimal million is that?
It's about 36.5 times lower than that 1 million per microliter minimum for PRP.
Wow. Okay, my head's spinning a bit. So based on the actual lab measurements.
Based on the actual lab measurements that Sasaki himself commissioned, the treatment used wasn't platelet-rich plasma at all. It was platelet-poor plasma, TPP.
So the poor clinical results, the lack of statistical significance.
Suddenly makes a whole lot more sense, doesn't it? If you're essentially treating patients with plasma that has fewer platelets than their own blood, you wouldn't expect dramatic hair growth.
It's like the emperor literally had no platelets. Or very few, anyway.
That's exactly the point Dr. Yam was making, and it gets weirder.
Oh, there's more.
Dr. Yam pointed out other odd things in the data presentation. For instance, Sasaki described both batch A and batch B as being 4.5 times baseline.
But wait, weren't they supposed to be low and high concentrations for comparison? How can both be 4.5x?
Good question. It contradicts the study's own premise of comparing different concentrations. And maybe even stranger, Dr. Yam noted that for both the average platelet count and the standard deviation, the reported values for batch B were exactly double the values for batch A.
Exactly double, down to the decimal point.
Well, the paper reported means and standard deviations, and the batch B values were precisely twice the batch A values.
That sounds unlikely if you're measuring separate real-world biological samples with inherent variability.
It's highly improbable, let's put it that way. It raises questions about how those batch B numbers were derived or reported, especially given the CCA seemed to only analyze batch A properly, if you follow.
Okay, so Dr. Yam's recalculation throws the whole study's premise into doubt, based on its own data. What about the manufacturer, Eclipse? Did this change anything for them? Are they still claiming 4.5x?
Well, according to Dr. Yam's letter and checking around the time he wrote it, yes, that 4.5x PIF value was still appearing on Eclipse's website for the system used.
Huh. Even after this critique.
It seems so, and this prompted Dr. Yam to ask some very specific, pointed questions about Eclipse's claims.
Like what?
Well, for one, Eclipse apparently claimed the PIF improved from 3.5x to 4.5x simply by using 2 of their identical collection tubes instead of 1, with no change in the actual processing protocol.
How would just using a second tube increase the concentration factor? That doesn't make intuitive sense.
Dr. Yam essentially asked the same thing. Where does the extra concentration come from? And then there's the math on the total number of platelets they claimed you could get. Eclipse advertised, say, 5 billion total platelets from their 22 milliliter kit and 10 billion from their 44 milliliter kit. They also claimed a platelet recovery yield of over 80%.
Okay, yield meaning the percentage of platelets from the original blood sample that end up in the final PRP.
Right. But Dr. Yam ran the numbers. Based on typical starting blood counts, to actually get those 5 billion and 10 billion platelets, you wouldn't need an 80% yield. You'd need yields of 93% and 123%, respectively.
123%? How is that even possible? Where do the extra 23% of platelets magically appear from?
Exactly. You can't get more platelets out than you put in. It points to a fundamental inconsistency in their own advertised numbers.
This seems to suggest maybe the issue isn't just with this one study, but potentially with the system itself, or how it's marketed.
Well, it certainly raises questions, and it ties into a broader point about the PRP landscape in general. There is a huge amount of variability between different PRP preparation systems out there. It's not all created equal.
How much variability are we talking?
A lot. One comprehensive review looked at 34 different commercially available systems. It found a massive 28-fold difference in the platelet concentrations they actually produced, from the lowest to the highest PIF.
28 times difference. So one system might give you barely any concentration and another gives you loads.
That's the reality. In that review, for example, the Eclipse system came out near the bottom, around 79,000 platelets per microliter in that independent test.
79,000. Oh, way below a million.
Way below. Compare that to something like the Arthrex system in the same review, which yielded 2.3 million platelets per microliter. Huge difference.
Wow.
And remember, the review also found that single-spin systems, which is what Eclipse uses on average, only produced a PIF of about 1.25x. Again, far from 4.5x.
So the type of system really matters. What about how it's used? Like the spinning, the centrifuge?
Absolutely. The settings are critical. The Eclipse system uses a single spin, and it's quite a hard spin, relatively high force and long duration, like 10 minutes of 1,500 times gravity.
Okay. Is that good or bad for getting platelets?
Well, independent research suggests that you actually get the maximum platelet yield, the highest number of platelets recovered at much lower centrifuge settings, like maybe 900 grams for 5 minutes or even 160 gram for 10 minutes.
The spinning it harder and longer doesn't necessarily mean more platelets in the end. That seems counterintuitive.
It does, but think about it. If you spin too hard, you might actually damage the platelets or pack them down so hard against the bottom with red and white cells that you can't easily collect them in the plasma layer. So, while a harder spin might give you a purer plasma sample, meaning fewer red or white blood cells mixed in it, can actually lead to a lower overall platelet concentration.
I see. Purity versus quantity.
Exactly. And those findings that higher forces lead to lower yields are completely consistent with independent studies measuring the actual output of the Eclipse system. It tends to produce lower concentrations.
Okay, so we've gone through the weeds of the numbers, the recalculations, the manufacturer claims, the centrifuge settings. Let's pull back. If you boil down Dr. Yam's entire analysis, what's the big picture message here?
The core message, I think, is Dr. Young's conclusion. You simply cannot blindly accept a claimed value, like a high TIF, at face value, especially not without independent verification, and especially when that claimed value seems to contradict other known research or even the study's own underlying data, as was the case here.
So the study wasn't really testing PRP as intended.
He argues quite convincingly that it was essentially a study on platelet-poor plasma, PPP, and drawing conclusions about PRP from a study using PPP is just, well, it's fundamentally flawed.
And this matters to anyone listening because
Yeah,
why should we care about the PIF of some hair loss study from a few years back?
because it's a perfect, if perhaps extreme, example of a much broader principle. It really hammers home how vital it is to scrutinize the methods and the actual data behind scientific claims, particularly when you're talking about new treatments, new products, things people are spending money on, hoping for results.
Yeah.
It shows that the details, how something is made, how it's measured, aren't just minor technicalities. They can completely change whether a treatment is actually effective or even what the treatment is.
It's a call for critical thinking, basically. When you read about a study or a product claim,
Exactly. Ask questions. Look for independent verification if you can. Understand that numbers, especially impressive sounding ones provided by manufacturers, sometimes need a second look. A small discrepancy on paper can mean a huge difference in reality.
and it probably adds weight to Dr. Young's critique that he declared no conflicts of interest, right?
Yes, that's an important point he included. He explicitly stated he has no financial ties to any company involved in PRP. That really supports the idea that this was an independent, data-driven analysis, not motivated by any competing interest.
Okay, what a fascinating deep dive into the numbers behind the claim. The main takeaway seems crystal clear. You absolutely have to understand the details, like the actual concentration of whatever's being tested, to know if the results mean anything at all.
Couldn't have said it better. The devil really is in the details, or in this case, in the platelet count.
But leaves you wondering, doesn't it? In this world where we're just flooded with information, new studies, breakthrough claims, how many other things that we take as established fact, how many other breakthroughs might look completely different if someone took the time, like Dr. Yam did, to meticulously re-examine the raw data underneath, instead of just, reading the summary and accepting the conclusion.
It's a sobering thought.
Makes you wonder how many other emperors out there might be, well, platelet poor. Something definitely worth thinking about.